The human body is very complex. It has to do a million things at once to make it through the day. In most cases, we ordinary students or even experienced scientists cannot fully imagine all of the millions and trillions of processes in our bodies. The most exciting part is to notice such little details as the molecule I will describe in this video and its huge impact on our lives. My nomination for Hero of the Year goes to N-methyl-D-aspartic acid. Its basic characteristics, such as melting and boiling points, stability in the air, flammability, toxicity, and others, are presented on this and the following slides. It is considered stable, it burns but flame with difficulty. It can provoke convulsions or have an effect on seizure threshold. Here you can see the hazard ratings. N-methyl-D-aspartic acid acts as a specific agonist at the NMDA receptor, mimicking the action of glutamate, the neurotransmitter which normally acts at that receptor. Unlike glutamate, NMDA, or N-methyl-D-aspartic acid in other words, only binds to and regulates the NMDA receptor and has no effect on other glutamate receptors, such as those for AMPA and KNET. NMDA receptors are particularly important when they become overactive, so does the N-methyl-D-aspartic acid, during, for example, withdrawal from alcohol, as this causes symptoms such as agitation and sometimes epilepsy. N-methyl-D-aspartic acid is an aspartic acid derivative. It has an N-methyl substituent and deconfiguration. Aspartic acid and derivatives are compounds which contain an aspartic acid or a derivative, thereof resulting from reaction of aspartic acid at the amino group or the carboxyl group, or from the replacement of any hydrogen of glycine by a heteroatom. N-methyl-D-aspartic acid is an enantiomer of MAAA. You can see it on the number 4, and selective agonist at the N-methyl-D-aspartic acid subtype of excitatory amino acid receptors. The standard agonist NMDA has R configuration and is more potent than the S enantiomer. This is opposite to what is observed for the enantiomers of glutamatic acid and aspartic acid. Another unusual feature concerning N-methyl-D-aspartic acid is its high potency in spite of the N-methyl substituent. N-methylation of other NMDA agonists normally results in a decrease in their activity. The most interesting part is the history of the molecule and why I consider it as a hero. So synthesis and study of N-methyl-D-aspartic acid and various NMDAR antagonists by Jeff Watkins and colleagues made a significant contribution to the discovery of NMDA receptors. These NMDAR antagonists were shown to block neuronal responses to applied N-methyl-D-aspartic acid, but not to block responses to KNET or quisquelate. As a result, NMDARs were shown to represent a distinct subpopulation of excitatory amino acid receptors. Over the next several years, these and other NMDAR antagonists led to the discovery that the NMDARs play key roles, so does the N-methyl-D-aspartic acid, in synaptic transmission, synaptic plasticity, learning and memory, neuronal development, stroke, seizures, and many other physiological and pathological processes. These studies generated great excitement about the potential use of NMDAR antagonists and synthetic N-methyl-D-aspartic acid to treat neuropathological and neurodegenerative diseases. However, the development of NMDAR targeted therapy has been disappointing. Several agents failed in clinical trials due to adverse effects or lack of clinical efficiency. But despite this disappointment, NMDAR therapy and synthesis on N-methyl-D-aspartic acid continue to exhibit significant potential. Of the multiple drug-binding sites on the various NMDAR subunits, many potential types of NMDAR antagonists exist, and some of these reveal distinct patterns of cell activity. Advances in our understanding of the pharmacology and function of the N-methyl-D-aspartic acid are likely to lead to the development of selective antagonists for this subunit. The combination of subunit selective pharmacological tools for NMDARs and molecular biological methods will provide significant information about the function of NMDARs and the role played by the individual subunits in the central nervous system. These advances are likely to open new possibilities for treating a range of central nervous system disorders, in which N-methyl-D-aspartic acid plays a significant role. So, due to the significant contribution to medicine and further development of science, we can consider N-methyl-aspartic acid a hero. Another part which I would like to talk about is hormonal release regulation. N-methyl-D-aspartic acid is implicated in hormonal release regulation. Synthetic N-methyl-D-aspartic acid provokes very strong activity for the induction of hypothalamic factors and hypophyseal hormones in mammals. Many studies have shown that N-methyl-D-aspartic acid is able to stimulate the release of several hormones from hypophysis gland. Hypophysis gland regulates growth, reproduction, metabolism, stress response, and lactation. So this small molecule plays a vital role. It allows us to live, in other words. In addition, NMDA receptors are colocalized in specific hormone-secreting cells of the anterior pituitary. The primary site of action of N-methyl-D-aspartic acid is at the level of hypothalamus via the control of hypothalamic releasing factors. Other studies revealed the presence of NMDA receptors in hypothalamic neurons and their association with gonadotropin-releasing hormone neurons. As for the details, I think that it's important to say that gonadotropin-releasing hormone neurons control the release of reproductive hormones, and the anterior pituitary is the front lobe of our pituitary gland. It is a part of our endocrine system, and it controls the function of several other endocrine glands. So the small molecule N-methyl-D-aspartic acid is responsible for many functions of our body. And the last part, which I would like to talk about, is two sides of the same coin. It is the side effects and benefits of synthetic N-methyl-D-aspartic acid. So, in adults, adverse effects of healing, of treating different disorders with synthetic N-methyl-D-aspartic acids are mainly central nervous system side effects, including hallucinations, lightheadedness, dizziness, fatigue, headache, out-of-body sensation, nightmares, and sensory changes. That's a lot, but it has also its benefits. So enhancing NMDA neurotransmission with synthetic N-methyl-D-aspartic acid can improve memory and behavior systems of both dementia and schizophrenia. And N-methyl-D-aspartic acid produces consistent, rapid, and sustained antidepressant effects in patients suffering from treatment-resistant depression. However, NMDA-induced cognitive impairments remain a major concern and still requires further research. Though the material on slides may sound complicated, N-methyl-D-aspartic acid can be considered a hero of the YAM without any doubt. I hope you enjoyed the material and you're now interested in further research on NMDAR receptors and N-methyl-D-aspartic acid synthesis. Also, you can find the list of sources used for this project in the description to this video. Thank you for your attention.